una grande fortuna, se posso dire così. Abbiamo qui il professore Gerry Melino, che è uno dei più grandi oncologi preclinici che abbiamo in Italia, e egli veramente ci fa questa grande sua liberalità, diciamo così, di venire qui a tenerci il seminario. È stato già alcune altre volte in quest'aula parecchi anni fa, e il suo progresso nel campo dell'oncologia, ma anche nel campo di alcune, se posso dire, monumental review che seguono a parti sperimentali in altri campi, come per esempio l'aterosclerosi, e come alcune altre review. Ce n'è una monumentale adesso sulla cell death, che è fatta da un gruppo molto numeroso, vero Gerry, di scienziati europei, quasi tutti, qualcuno anche americano, proprio su questo grande capitolo dell'apoptosi, che era fino a 30 anni fa quasi sconosciuto. Gerry Melino è napoletano, nato a Napoli, al centro storico, credo, vero? Al centro storico, e poi però è andato via da Napoli a pochi anni di vita per la famiglia, naturalmente, che ha seguito i genitori ed altro, ed è in questo momento è membro dell'Accademia dei Lincei, la migliore accademia scientifica culturale italiana, della European Academy Cancer Studies, questa accademia europea, e è professore da pochi giorni emerito dell'Università Tor Vergata di Roma, di biochimica, proprio di biochimica, anche se è stato già professore di biologia molecolare, poi giustamente si è stufato un po' ed è passato alla biochimica. Scherzo, perché quasi sono i biologi molecolari e facciamo sempre questi scherzi. Subisci, che qualche volta. Allora, poi egli è stato direttore di un centro di eccellenza sull'oncoscienza a Tor Vergata, ed è stato per 19 anni direttore di un cancer program in Inghilterra, del Medical Research Council a Leicester e poi anche a Cambridge, e quindi è stato anche direttore di un laboratorio a Pietroburgo in Russia. Quindi vedete un poco, io lo chiamai una decina di anni fa, dico: vuoi fare un gruppo, un laboratorio a Napoli, nelle cose? Io faccio già troppe cose, e non potemmo avere questo onore e questo privilegio di averlo qua come direttore di un gruppo di ricerca in questa struttura. Egli ha creato un forum, diciamo così, per la discussione e l'innovazione di un grosso capitolo, quello che dicevo prima, cioè della morte cellulare. A un certo punto il professore, in una bellissima opinion paper, o anzi credo position paper, sì, forse è meglio chiamarla così, e ha dato molto vigore a questo campo di studi, e ha fondato ben 3 riviste scientifiche di alto livello, che sono una Cell Death and Differentiation, un'altra Cell Death, un'altra, diciamo, che comunque parla sempre di questo capitolo, che hanno anche un ottimo impact factor, e degli editor in chief. Dicevo che in questo campo, alcuni anni fa, credo una ventina però, egli scrisse un articoletto, di tipo position paper, stavo dicendo, intitolato Il canto delle sirene, una cosa del genere, molto grazioso. Sta su Nature, un editoriale in cui parlava, mischiando la mitologia greca al problema della cell death, e in quella occasione io mi ricordo che incominciai a capire quanto era importante questa cell death, perché se non esistesse questa cell death noi avremmo quante tonnellate, mi pare, non lo so, diventeremo, se le cellule non muoiono e si accrescono sempre, noi diventeremo delle specie di, no di balene, ma molto di più delle balene. E quindi questo è un campo che stranamente fino a 25-30 anni fa era poco individuato, poco studiato. E poi avremmo, non lo so, che lunghezza, quanti metri, quanti chilometri di, 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 di intestino, avremmo, non lo so, anche di pelle, una quantità molto come metri quadrati di superficie, quindi una cosa sfiziosa a rifletterci un momento, non solo come curiosità ma soprattutto come scienziati. Bene, il professore ha avuto un sacco di premi, il premio maggiore dell'Accademia dei Lincei, il premio Feltrinelli, che è presentato dal Presidente della Repubblica Italiana, il premio Antonini, un grande studioso della nostra biochimica, uno dei maestri fondamentali nel campo della struttura delle molecole, in particolare dell'emoglobina ed altre, e poi dottore di scienze in varie città, diciamo dell'Italia, ma anche awards addirittura in Cina, oltre che in Russia, nelle Hawaii, eccetera. Ebbene, questo training e questi meriti del professore Melino sono molto utili ad essere detti, non perché io dovevo fare 5 minuti di chiacchiere, ma perché credo che i giovani è bene che sappiano quale carriera, quali sforzi, quali metodologie sono stati usati, e il professore ne è diventato, se posso dire questa parola, un principe, e ha incominciato a lavorare sulla morte cellulare della pelle. Siccome oggi gli parlerà, credo, della regulation dell'epithelial cell fate, ecco che lui ci rappresenterà non studi dell'altro giorno, ma studi forse che si sono sviluppati in un periodo abbastanza lungo di tempo, e quindi c'è una storia. Quando c'è uno studioso riesce a fare una storia su un argomento, credo che sia una cosa molto bella e molto importante per la carriera dello stesso, ma anche per la scienza stessa. Grazie di essere qui, Gerry.
Devo parlare in inglese o in italiano?
Dunque, se c'è qualcuno che non capisce l'italiano, allora il professore gentilmente parlerà in inglese. Noi abbiamo la regola che qui bisogna parlare in inglese, poi viene poco forse adoperata, ma in questo centro, diciamo, si dovrebbe sempre parlare in inglese. E allora grazie Gerry, se tu parli in inglese siamo tutti molto grati.
Grazie a te. First of all, I would like to thank Professor Franco Salvatore because we've been friends for a really long time, and I think I've seen in the last 20 years at least the growth of this institute, and I've seen how this institute has been going to the top level in Italy and a center of attraction worldwide. So I am particularly pleased also because now this institute is called dedicated to Franco Salvatore, and I think at the personal level this is really a great achievement for a scientist who has established a center of excellence, but it's also a way for recognition for what you have done. So I'm particularly pleased to be here because I've been here twice before, but this time I'm at the Franco Salvatore Institute, so for me it's a huge pleasure to do this. Before I start my seminar, I would like to disclose my collaboration in Germany. I have two in Germany, and of course what Franco said is also the collaboration for these three journals in which I'm deeply involved. They all related with Springer Nature. The first one of them was established 30 years ago, and I have to say for the young scientists, you know, don't be afraid of trying something crazy or outrageous, because you know, I would have never believed after 30 years to be here and present things related to the journal. Of course, normally, because I'm related with these three journals, I never present things which have been published in these three journals for obvious conflict of interest. But when I was younger, don't afraid in doing whatever you like to do. If you think that a lot of people who are now celebrated, like the founder of Microsoft, or of the founder of X, Twitter, or this center, they've launched this initiative in their 20s. So don't be afraid of trying something unusual, something complicated. I also I sent a small preliminary of what I was going to say to Franco, and I was in that abstract. I was talking about P73, P63, and a little bit of transglutaminases. And I wrote a phrase in Napolitan, aviden azzerrati cash. Is at the end of the day, life is like closing the coffin, closing things together. Therefore, having written this, I basically would like to give my talk upside down and talk about the death, talk about the cash, about the formation of the conifer envelope. And here I'm talking about the skin. Grazie. Oh, thank you. Sì. And just very, very briefly for the people who are not acquainted with the skin, there are 4 different layers in the skin. The basal layer is laid down on a basal lamina, and this is the proliferation area, and then there is a differentiation and death. So at the end of the day, basically, the keratinocytes are death. And the second reason, because I want to talk about that first, is because normally we transport words and therefore concept and ethics from one area, which is, for example, philosophy, ethics, into another area, which is science. And therefore this brings unusual concept which maybe are wrong. So we normally think that after death there is nothing, and life is what's happening here in the basal layer, and what happened after is death. But this is not true for at least for keratinocyte, for red cells, for other cells. It's because the function, the real activity of a keratinocyte is after death. So there is no, in a way, if I exaggerate, there is no role for a keratinocyte, no function for a living keratinocyte. The first thing the keratinocyte does in this area is, there is a pointer. Okay. The first thing is basically to lose a couple of things which are fundamental. They lose the nucleus, therefore they lose DNA, and they lose the mitochondria, so the ability to make energy. So at that point, the keratinocyte is, in inverted comma, death, because they is not compatible with life. But still the differentiation goes on to form what basically is a coffin, a cache, which is where the function is exerted. So there are different ways of dying for a keratinocyte. I would say in simplified terms that there are 2 different types of death. One is the formation of the conifer envelope, and the other one is a programmed cell death. It's not one type of cell death, because these are known as sunburn cells for the dermatologist, and requires a classic apoptosis. But there is also RIP kinase 1, RIP kinase 3, MLK1, which basically are involved with the necroptosis. And there is also glutathione 4, which basically, glutathione peroxidase 4, which basically indicates that there is also an essential component of ferroptosis. So different type of programmed cell death can occur on the basal layer. But the important death for a keratinocyte is basically into the upper layer. The death is what I'm saying in Napolitan, inserrare cache, so closing the coffin. And basically it's the formation of a basically an empty box with a lot of proteins and a lot of lipids, which basically is the function of the skin. So the formation of this conified envelope is essential for a precise death. So in order to actually allow what people would call differentiation, I'm just calling preparation today, is basically avoid a classic programmed cell death, because you don't want death. You want a specific way, specific order in order to generate this death. So the proteins and the enzymes which are expressed in the skin are very strange. Here there are some, loricrin, profilagrin, I was too fast. Profilagrin, desmoglein, desmocolin, and a lot of enzymes, transglutaminases. Transglutaminases, they made cross-link between two proteins. There is a transglutaminase 1, transglutaminase 2, 3, and 5. So there are a lot of different transglutaminases, and with Franco we were talking before about one of these transglutaminases. And the proteins are very strange. Very strange in the sense that if we can see the amino acid composition, loricrin has 60% of the amino acid is only one amino acid. For biochemistry, this is insane. You have 60% of the sequence is only glycine. 24% is only serine. So basically, more than 80% of the protein is made by two amino acids. There are no proteins that exist in this way. But if you look into the other proteins, you see keratins, they have 23% glycine. Filagrin, again, profilagrin actually, is 15% or 20% serine. So the concentration of unstructured amino acid is huge. And then look at this. These proteins, SPR, has 43% of proline. 43% of protein. Normally, if you have one proline, 3 protein in a protein is significant, but 43% is impossible. What these amino acids have in common? Glycine, serine, and proline, they are, sorry, it's my finger. They are basically unstructured. There is no alpha helix that is compatible with this structure. So these proteins are completely unfolded. I'm putting here the person who introduced me to this biochemistry, Peter Steinert from NIH, and we've done a lot of work together with Peter, and then we continue by ourselves. So what is the logic of creating this coughing of cells? First of all, the skin is not plastic. The skin needs a structural stability. The skin needs elasticity, because otherwise it would be like wood, like plastic. The skin needs also mechanical resistance and water repellence. So these are the physical properties which are important for the skin: structural stability, elasticity, mechanical resistance, and water repellence. And we can link this property to cross-linking enzymes, to the substrate of these cross-linking enzymes, to the cytoskeleton of the dead cells of the coffin, and a complex lipid structure which is essential for the skin. And this leads to an investigation of transglutaminases. I already said there are 4 different types of transglutaminases in the skin, not 1, 4. Why? And we will see in a moment, because the way they have to die is very, very specific. And then the subset I already mentioned, small proline-rich proteins, proline-rich, 43% of protein in the sequence is unbelievable, but also the other one, they are completely unstructured. The cytoskeleton is related to keratin, and lipid attachment to, this is a fatty acid, long fatty acid dehydrogenase. And this leads, abnormality of this sequence, it leads to diseases. There are, I was just mentioning before, with Professor Esposito, a collaborator of Franco, that there is a ichthyosis, some diseases related to a specific transglutaminases. There are several substrate abnormality like Vovinkel. I don't know why I brought Vovinkel. There is at the moment, as far as I know, there is a 0 patient in Italy on Vovinkel. So, but there are other diseases. Maybe, anyway. And then there are a lot of keratin diseases, Pachyonychia congenita, white spongious nevus, epidermolytic palmoplantar keratosis, and so on. And Shuren Larson was identified in my laboratory long time ago by Vincenzo De Laurenti and Peter Steinert. So there is a specific enzymes, a specific substrate which form that unique way of dying. The process of dying is basically elimination of the DNA, elimination of the programmed cell death. You don't want unspecific necroptosis of death. You want that kind of a way of dying. So there is an initiation by a transglutaminase, which is 5, and basically below the cell membrane start cross-linking the substrate. Then there is a prolongation, more cross-linking by another transglutaminases, consolidation, another transglutaminases with additional substrate. So the sequencing of what you build, what you cross-link with the next one is very, very delicate and specific. And then there is the lipid, like the FLDH and other enzymes. So biochemically, you can envisage that from the spinous layer, the granular layer, the upper granular layer, the conified layer, you have a sequences of transglutaminases, calcium-related enzymes with the formation of lamellar body and lipid, cross-linking on the desmosome. This is transglutaminase 1, this is transglutaminase 3, this is transglutaminase 5, and gradually you build up this coughing, which is the function of the keratinocyte. So what we have done in 20 years is basically characterization of the transglutaminases, characterization at the biochemical level. We have done the knockout of transglutaminase 2, transglutaminase 5. Nobody has done the knockout of transglutaminase 1 because the people that were thinking that we were doing the next knockout, which we didn't. We didn't because we thought was automatic to understand what is the phenotype, and therefore was difficult to publish. Then we spent some time working on the substrate of transglutaminases, and then what is the role in disease. This has been the rationale of what we were trying to do in the last 20 years. So I've just mentioned before that there is one disease in which we contributed to identify the cause, which is the mutation of transglutaminase 1 leading to a ichthyosis. Ichthyos in Greek is a scaly fish, is the skin of the fish, and this is the way they have the appearance of the abnormal cross-linking of keratinocyte. This is the enzymes are 2 beta. All the transglutaminases, they are similar. They have 2 beta barrel. They have a catalytic central domain. This is the catalytic cysteine. And then there is an N-terminal domain. And during the catalysis, there is a movement of the N-terminal domain over bringing a channel for the substrate to be activated, and all similar with the others. So mutation in TG1 basically leads to this genetic disease. At least in 50% of the lamellar ichthyosis, this is the cause. And there are still about 20% in which the cause of lamellar ichthyosis is not known. Phenotypically, it's the same. So let me say one more slide on the transglutaminase 1. Transglutaminase 1 has a molecular weight of 106 kilodalton, can be a soluble enzymes, or it could be bound by myricylation and palmoetylation, one or the other, but then at the end it's both. So the enzymes needs to be palmoethylated and myrcylated and membrane bound. Then there is a proteolytic cleavage of the transglutaminases, and therefore you end up with three different small protein of 10, 67, and 33 kilodalton. Again, could be attached by to the membrane or cytosolic. So this is to highlight how complex to be the function of the transglutaminases. But this is catalytically relevant, because if we measure the picomolar putrescine bound to picomolar proteins, we go from 5 to 50 when it's membrane bound or when it is soluble, and you could go up to 1,000 when it's membrane bound proteolytically cleavage. So the post-translational modification, lipid attachment, and proteolytic cleavage is extremely complex for transglutaminase 1. So let me say a few things on the substrate. The first substrate I'd like to say is loricrin. Loricrin is a very, very strange protein. This one has 88% of made of 4 amino acids, 80% of 2 amino acids, glycine and serine. Glycine is not compatible with alpha helix, with all the structure. So the protein by definition is unfolded. But the glycines is basically in three areas, 3 loops, which are called omega loops. And these omega loops, they carry also transglutaminases. They cross-link protein, but they cross-link a lysine with a glutamine. And lysine and glutamine can be in the middle of the omega loop or between one omega group and the other one. And if we measure the catalytic activity of transglutaminase 1 cross-link in this area, very little in this area, and a little more in this other area. Not this lysine is never used by transglutaminase 1. Transglutaminase 3 has a different site and different catalytic activity. So each transglutaminase uses different residue with different efficiency. So this is the structure which already said. Loricrin made up 82% of the conifi envelope. Of the dead keratinocyte in dry weight of protein, 82% is loricrin. It's amazing that there is one disease, that's why I call the Vovinkel disease, in which there is a mutation of the 5' end of the protein, and there is no expression of loricrin. And therefore, even though 82% of the dry weight of protein is normally is loricrin, these patients, they are badly compatible with life, but they are alive without loricrin. Means that the other substrate, they compensate in a huge manner. This is interesting. I only wrote it here because the forestomach of the mouse is different from the stomach of the human. This mouse, they have a keratinocyte skin like our external skin, and therefore the mouse not necessarily is a model for human diseases. Okay, having said that, going back to the same skin, basically we see that transglutaminase 5 seems to be the first in cross-linking, followed by TG1 and transglutaminase 3 in a very ordered manner. Very ordered manner. You cannot reverse this disorder. So this is the structure of the sequence, not the structure, of SPR. And on the bottom, we can see the NMR that we did some time ago of these 3 loops. indicated here. Basically, they are completely unfolded. They do not have a structure. They vary very, very rapidly. But what we wrote here is the ability to use glutamine or lysine from transglutaminase 1. So you can see that transglutaminase 1 is not able to cross-link, very, very little. Transglutaminase 2, 0 or close to 0. 3 has a certain selectivity for this lysine. But if you add first transglutaminase 1 and then 3, you can reach a significant activity. But if you do 3 and 1, first transglutaminase 3 and then transglutaminase 1, you reach 90%, 100%. This is the C terminus, this is the N terminus. Although there is a lysine here, in any combination, this lysine is not used for cross-linking. The protein they are cross-linked, in summary, at the N terminus, at the C terminus, never in between. So basically, this is what looks like. Transglutaminases, they cross-link at the N terminus, these lysines, and the N terminus. And although they have a potential substrate in the central domain, they're never touched by transglutaminases. Why we think this is happening is because if you cross-link at one side and the other side, then you have a flexible, unstructured loop in between. And therefore, you can allow to extend in like an elastic, the structure. So the resistance is given by a very solid cross-linking from at the two ends. But then the middle is elastic. It's like this elastic that you use for a motorcycle. You know, you create a net of cross-linking protein, but they are expandable, and you can expand and give what is a peculiar form of the skin, which is elasticity. So skin, again, is not plastic in which you cross-link everything. You need to cross-link a certain substrate in a certain order, and then you can gain elasticity. Cytoskeleton. The really structure, because this protein, they're not so able to form a really big network. So what happened is that the keratins are forming the cytoskeleton, and there are a lot of diseases that can be formed. In this was what spongiosne was identified in my lab by my people, and we thought was keratin 4. And at that time there was Irving McLean who published back-to-back that was keratin 13. But keratin 4 and 13, they form heterodimers. They join together. So whether you have the mutation of one homodimer, one component of the heterodimer, or the other, it doesn't matter. You have the same disease. And then we identify a very, very unusual situation in which you have this into homozygous. And that is being classified as a new disease. In OMIM, it is indicated as a new disease. Keratins, they have, and Caterina knows this, they have two linker regions, and what happens is that they form two chains. They form dimer, then they form tetramer, and then they form very long fibers. So this gives the mechanical resistance. For each of the keratin, you have there is a plethora of diseases here, and depending of which keratin, this is epidermolytic palmopalmar keratosis, you have a different keratin, epidermolysis bullosa simplex. Michele De Luca has done a lot of work on this. And you have a lot of diseases. The majority are on the domain 1A and 2B, because those are the domain important for the cross-linking. So what we have done in the past in this area, what is still continuing, is to understand the function of keratinocyte is after death. And they need to have stability, mechanical resistance, elasticity, and water repellence, thanks to two classes of things: the enzymes cross-linking and the substrate, which are very, very unique. They are not in other, and this creates the barrier. And then mutation in either the enzymes or the substrate basically create diseases. I'd like to shift now to what has become the interest of my laboratory, which is not female fidelity, but is the germline, the germline control of ovocyte. This was published 2 years ago, so I just very briefly want to mention this. So, what is the question? What is the, what are we talking about? So in the ovocytes for female and for humans are essential. And at birth, there are 7 million ovocytes. Immediately after birth, you are reducing this to 50%. So 3.5 million ovocytes, they died immediately after birth. Immediately means in the next 2 days. And this is the post-Pakitan checkpoint, is called. And then they gradually reduce the number of ovocytes until puberty. The girls at puberty, they have already less than 500,000. So you'd go from 7 million to 500,000. Then you need in the lifetime the ability to have kids, 1 every month, ovocytes. So maybe you need 200. And then at menopause, they are completely gone. So why you have such an inefficiency? Why, in order to have maybe 1 kid, 1 ovocyte, you need 7 million? So this is because there is a huge quality control on this ovocyte. And the quality control is unbelievable. It's probably one of the strictest control, which allows us to have only 2 hands and 2 feet and not 6 and genetic abnormality. So the clue in this came from a friend of Caterina, a friend of myself, with whom we collaborated, Frank McKeon. Frank McKeon showed that the P63 is the gene on the primary, secondary, tertiary ovocytes, and in the nucleus is full of P63, specifically on TA P63. It's one isoform of P63. So there is no P73, there is no P53, but this gene is highly expressed from the birth, before birth, till the menopause. So this protein is there for 50 years, the same protein. Why did I say P73, P73? It's because this protein, P63, belongs what is called now the P53 family, which includes, beside P53, also P63 and also P73. So there are 3 genes. Strange enough, they all have 2 promoters. With a lack of imagination, we call it promoted 1, promoted 2. We could have called it Giovanni and Alberto, but that's okay. And they all have 2 promoters. and they codify protein from one promoter or from the other one. We call it TA because this is the transactivation domain. We call it TA, the one codified by the first promoter for both P63 and P73. Delta N because it's a short at the amino terminal, the use of the second promoter. So the gene is quite complex. It's quite multi-exons, and there is a huge gap between promoter 1 and promoter 2. I don't know why there is no evidence, in my view, of a single promoter. It's always 2. So there is talking about P63, there is a very specific expression of promoter 1 only in the ovocytes. There are very, very rare exceptions to this. And promoter 2 is the one used in epidermis. So in the ovocytes, it's only TA, only promoter 1, tissue specific, of course. And the second promoter is only in the skin. So we have noticed we have done a knockout for all these members, TAP63, TAP73, Delta NP73. What we published a couple of years ago, we've done a selective knockout of one exon, exon 13, which codify the sterile alpha motif. It's the only structural motif which has been structurally identified in this area. And interestingly, technically is a knockout. Functionally is not a knockout because the protein is expressed with the right promoter in the right place at the right time. So there is only one thing, is that instead of producing the alpha isoform at the 3' end, there is a switch from alpha into beta. So this is functionally is like a knock-in. You substitute the alpha isoform into the beta. So the question of this is basically to identify not what is the role of the 5' end of the promoter, but of the splicing isoform. So the mouse has basically, when it's recombinant, even in the heterozygous, has basically only beta isoform instead of alpha. The mouse is more or less normal. The abnormality comes here. I'm talking about the heterozygous, not the knockout. The heterozygous, the fertility of the man is absolutely normal. The female, they are not even one pup. So it's completely infertile. So this demonstrates that the quality control at the evolutionary level between man and female is completely different. And the quality control is exerted by TAP63 in the female, not in the male. So we analyzed MYS, which identify primordial oocyte. This is the wild type, and this is the heterozygous. And we can see that in the heterozygous, there are very, very few oocytes in the ovary. And after postnatal day 5, postnatal day 10, basically there is no oocyte whatsoever. They're completely gone. They completely disappear. And of course, P63 was absent in the heterozygous, but there are no oocytes. So we crossed this mouse with the c-kit, because the c-kit stains with the GFP c-kit, because so we can actually visualize better, and the oocytes are green, and c-kit identify primordial germ cells. And if we analyze, these are heterozygous, and these are the wild type, postnatal day 1, 2, 3, 4, we can see in the wild type what I've described at the beginning. Within a few days, 50% of the oocytes, they have gone. They die at birth immediately. whether it's oxygen, whether it's hormones, I don't know. But this is a very well described mechanism. There are very few ovocytes into the heterozygous. After 2 days, there is none. All the ovocytes, they are dead, and they are died because of the classic apoptosis. There is the induction of PUMA, induction of NOXA, and so on. We can explant primary ex vivo ovocytes, and we can stain with the, they are green because there is a GFP kit, and we can stain for cell death. So red dead cells, ovocytes, green is ovocytes, and color, these things here, are supporting cells. No? And if we stain for the heterozygous, you see how red they become. These are two different pups, sibling. You can see that they die. As soon as they're explanted, they die. What happened is that, with a collaboration with Volker Deutsch, we've seen that the isoform alpha is the dimer. This is unusual because P53, which has been discovered in 1979, is a tetramer, but P63 is a dimer. But the isoform beta and gamma, the other two isoforms identified, they are tetrameric. And interesting, the alpha, which is dimer, has a very weak transactivation ability over PUMA, cell death, anything you test. But the beta and gamma, the tetrameric isoform, they are basically transcriptionally active. So what happens is that when you have a genotoxic damage on the ovocytes, chemical or physical, basically there is a ATM and CHEK2 phosphorylation on the dimeric isoform of P63, which then leads to opening of the dimeric interface. You have a casein kinase 1, another kinase that can keep phosphorylating and opening these arms completely, and the disassemble of the dimer forces only one reassembling, which is tetramer, and therefore the protein goes from dimeric to tetrameric, and therefore the tetrameric is transcriptionally active, and therefore you can transcribe the target, the cell death targets. So basically it's a switch physiologically from dimeric to tetrameric, which allows the killing. This also open potential therapeutic venue, because you can block these kinases in order to prevent the killing, the cell death. And you can simulate in vitro and simulate chemotherapy in vitro, and again red is the dead cells, and you can see that after damage of cisplatin 20 macromolar, the cells, they become completely dead. So they are super sensitive to death. So imagine that this is a woman undergoing chemotherapy, for example, for a breast cancer. A side effect of the chemotherapy could be infertility. So with Stefania Gonfloni, we treated with cisplatinum, and these are primordial ovocytes. They are completely gone. And P63, which is here in red, they are not primary keratinocyte, it's oocytes. And therefore, imatinib doesn't do anything, but if you deal with the cisplatinum, the number of the black bar is very, very low. So if you block the kinases, you still have primordial ovocytes, and you still have P63, so you preserve the integrity of the ovocytes. So imagine to exploit this to a woman undergoing chemotherapy. If you block that system, you basically allow the oocyte to survive. Not only for kids, but also for the hormonal function of the oocytes. And in mice, basically the pups after chemotherapy with imatinib basically recover, and they actually make normal pups. I wouldn't do it for human, but anyway. So interestingly, in cancer, there is overexpression of a delta N p63, which is the other isoform, and in the ovary is the TA. So by blocking, you don't have to have a problem of reducing efficacy of the chemotherapy, because you can preserve, in theory, the integrity of the ovary function while attacking the tumor. In theory, because that needs to be tested. So another group has identified MP63 a mutation not only for the genetic diseases that Caterina is working on, but in primary premature oocyte insufficiency. So this lady are infertile, and the first paper that I'm referring to, there were 13 ladies with infertility, inability to have at the fertility clinic to have kids, because they had mutation, guess what, in the same domain that we eliminated. We tested a lot of these mutants, and the ability to transactivate the targets is all the mutants, they are very powerful in transactivating. The scale is 100-fold the transactivation, so it's very low, but it's a very strong transactivation. And we checked with Volker Deutsch also whether it's dimeric or tetrameric, and interesting, they are all tetrameric. So the disease is there. The problem is that you switch from a transcriptionally inactive isoform dimer into a transcriptionally active and therefore killing isoform. And this, instead of the dimeric allowing a normal oocyte, basically the tetrameric isoform by mutation or by damage basically depauperates from all the follicle from the ovary. Okay, and it's saying that I'm conclude that the SAM domain, that domain is essential for the function of the AP63, and also indicates that the alpha isoform is essential for the function. Beta and gamma, they're not. This is about integrity of the female. What about men? I don't know about men, but I showed here the what is called normal for the WHO for men, for the spermatozoid. So men are very macho, these things, but what is normal? And this is still valid. It is normal to have a sperm count in which 70% they are dead, they don't move, they are crippled, in which 40% are absolutely dead, and 97% they are strange looking. So this is not so good for the macho mentality. So thanks to the female that allowed us to keep the species alive. And they allowed us also to have this is a human case of blastocyst formation. So this was published, and we are now doing something else in the laboratory, which we have generated a because we cannot make the knockout because they are infertile. We had to make a keratin 14 dependent knockout. So the mouse is still under evaluation in the lab. We have not published yet. And a lot of the mice, they die, 40% they die. The mice, they are small, they fail to drive. They have abnormal skin. The skin is very, very thick. And as you can see, hypercondensed, the keratinocyte. So the normal cornification is not there. And there is a co-expression of upper protein and lower protein. They mix together. You have a green and red in these cases. You have basically keratin 14, which is supposed to be only here, and loricrin, which is supposed to be only there, and you have the mix of the two. So, and the basal layer is an hyperproliferation. We have done RNSX, a lot of cell adhesions expressed. So also for the skin, the alpha, the STAM domain is essential, which means the alpha isoform is essential for the function. Beta and gamma, I do not have evidence. So we are at the moment doing RNA-seq on mouse epidermis, on mouse keratinocytes, and we characterizing the absence of hemidesmosomes, abnormality into the tight junctions, and which we can see morphologically in these mice. So this I hope to conclude maybe next year. So meanwhile, what we have done, we read a paper by Paul Kawari. I was discussing with Caterina just a moment ago, in which there is another target, an interactor, but target of a P63, which is called ZNF750, a zinc finger protein. So we have done the knockout of this 750. This has just been published, so I just fly over this. The mice, they are all dead, and they are dead within, this is hours, 9 hours. They are alive, and within, on the first day, they are all dead. And the skin is there, so the phenotype is not like P63. There is skin, there is formation of the skin, defective is abnormal, but it's still there. There is a permeability problem. This is the skin barrier with the zinc sulfone biotin is the inside out and also from outside in. In both directions, there is a lack of the permeability of the skin. The barrier of the skin is abnormal. The lipids, this is a glucosylceramide staining, is a okay. So the glucosylceramide and ceramide is completely missing from the upper layer and even in Nile red. And by morphology, ultrastructure, we can see that there is abnormal formation of the lamellar body and lipids formation. And this, they require processing of a cathepsin D, which is abnormal in the mice. To make the story at the biochemical level, from the basal layer to the conifying layer, there is a very, very complex lipid evolution from palmitoyl coenzyme A, sphingogannine, then to glucoceramide, sphingosine, and phospholipid. So there is a very, very specific way in which the lipids are formed. And in the mice, we can see that there is the sphingomyelinases. They are much lower than in the wild type. The DGAT, deacylglycerol acyltransferases, they are reduced heavily, and also DGAT2 is strongly reduced. And also the sphinganine chetoreductases are reduced. These are enzyme expression. So there is a reduction of expression of a lot of the lipid. All the pathway is abnormally controlled into the mice. And if we reintroduce, if we reintroduce ZNF750 into the knockout mice, we reconstitute deacylglycerol acetyl transferases, and we basically we can reconstitute the enzymatic level in the keratinocytes. And because of this defect, if we measure, for example, a very long 52 carbon residue of triglyceride, they are strongly reduced. You see the yellow in the knockout is much lower than the green in lower. So this is mainly due by acyltransferase ceramide, but also to a levanoid. A levanoid is a class of enzymes which elongates it does the opposite than beta oxidation. It extends the carbon 2 by 2 and up to formation of 48, 52. And the absence of the levanoid basically induces the absence of long chain triglyceride and the acylglycerol and also all the derivatives of this. This happens also in the human. So if we silence ZNF750, the same enzymes are in primary keratinocyte are reduced, and this is due to a binding site on all these enzymes. These are the levanoid 6 and 7 and 4. They have a binding site for ZNF750, and therefore the enzymatic activity of these enzymes is reduced. So basically, although was described as an interactor and target of P63, it works in a different way, because the P63 works in the basal layer, and these lipids, they work in the spinous and granular layer. So although they interplay, ZNF750 works completely independent and is responsible not for skin formation but for the lipid formation, the lipid barrier. And there is a genetic disease associated with this. Very, very briefly, I only have one slide to indicate, we published that 4 years ago, in which another knockout shows that elimination of elimination of the same SAM domain, the same alpha isoform in P73 gives a huge phenotype. This is hippocampal. I don't want to say anything more. That basically to indicate that this is general for the entire family. So the two promoters, they are important because the first promoter and the second promoter, they exert very, very different function. In the case of P63, the first promoter is related with the ovocytes, and the function is to kill. In the second promoter is related to epidermis, and the function is due to control the stemness potential, no, and the differentiation. But this, on the 3' end, there is a regulatory domain, and this is true for P63 and P73 everywhere. This is the regulatory domain, and therefore the alpha isoform for both family is essential. I have a big question mark of whether the other functions are functionally relevant. So this allows me to wrap up what I have to say in the last 20 years. And we did publish a review 10 years ago in which we indicated that P63 control the ovary formation. I've shown you that with the recent knockout. P73 controls the blastocyst proliferation. P53, Arnie Levine has shown that control the implantation of the egg. The entire family has a function related with basically with the fertility. And we like to believe that the first function of the protein is basically maternal reproduction. and control of the quality check in the fertility. And this is conserved for P53, P63, and P73 for the entire family. This model has been evolved to be adapted to epithelia and the fate of the control of the quality of the epithelia, and also deregulated in cancer. But the first evolutionary origin is in female fertility. In fact, there is not 3 but 1 genes in C. elegans, is called SEP1, and is basically identical in structure like P63. And in C. elegans, there is no MDM2. There is only H, which is the E3 ligases that control the stability of P63 and P73. So we can generalize even more. We could say that the origin is basically the control of the quality of the quality check of female fidelity. So the entire family derived from as an aim the control of the ovocytes. And in evolution, these evolved to P73 and P53 new genes compared to P63, and moving from quality check of the ovocytes of the reproduction to quality check of the somatic cell, and therefore the abnormality, which is tumor suppression. And interestingly, the evolution allows a dimeric control of the function of the protein to a tetrameric control. The dimeric is inactive. That protein has to stay on the ovocyte for 50 years. And maybe can function instantaneously in any moment. But for P53, when there is abnormality, when it's expressed, has to kill straight away. There is no waiting. So very high activity, it's already a tetramer, very, very low concentration kills. P63 is different. They need to be tetramerized and so on. And there are in this pathway potential way of intervention. So conclusion, the epithelia, the cells, the keratinocytes are born to die. They are born with a P63. They die with a transglutaminases and their substrate, and they need a lipid regulation as a part of the function. The isoform alpha, which contains some domain, is essential for P63, P73. And P53 is an evolution of P63 from female germinal fidelity to tumor suppression. And okay, I can summarize also in another way. We live in a difficult environment. We have a difficult moment, and men are not so good in controlling this quality control. I have to thank Frank McKeon. I have to thank the people from my laboratory, Eleonora Candy, One for All, then Volker Deutsch, who's a long-time collaborator for me, and also with Caterina, Frank McKeon, the same, Takmak, the same. And then, but I have to thank this guy, JBS Alden. JBS Alden, he doesn't know I'm thanking him because he's dead. And he wrote a paper in which he says there is an extremely low contribution of mutation in the female germline. And we would like to think, Frank, myself, I guess Caterina, and also Volker, that this unknown factor is P63. Just as a question of curiosity, guess what? There were problems in the Middle East and in Israel and Sinai and Egypt. And JBS Alden was so upset that he left UK, he burned his passport, went to India and became an Indian citizen, and he died in Bhubaneswar as an Indian. And he had the time, since the Suez problem and his death, to actually establish a very, very good, high quality genetic institute in Budermasvar, which is still near Calcutta, which is still very active and very prolific now. So a big thank you to JBS Alden. Thank you.
Thank you very much, Gerry, for this beautiful lecture and for having synthesized for us 3 lines of your beautiful work in the, let's say, 20 years or maybe even more in some cases. And I take a little advantage of being here to make the first question, a very simple question, and I would like to understand if there is an aging possibility of calculating the, at least for ovary or maybe also for other organs, the aging process by diseased situation. So to make a kind of association or maybe a cause effect mechanism by which you can, because this is a beautiful, I think is a beautiful model that we can try to get information about this phenomenon, which I think is due, phenomenon of aging, which I think is due to alteration of molecular and cell alteration. Yep. For further. And if I make a second question, very banal, is it just loricrin? Loricrin is why there is no many mutation, no many disease, just the Fowinkle disease, just because of the structure is so tight in a certain sense that is not affected by environmental, because there are mutation, germinal mutation, but there are also environmental mutations that can work out disease, molecular alteration bringing into diseases.
So for the second question, the brief answer, I don't know. Loricrin is probably, there are many sites of cross-linking of transglutaminases at the, let's say, at one side and the other one. So if you mutate 1 or 2 or 3 of these sites, can still be cross-linked by one of the transglutaminases and therefore give this elasticity. So probably not being related with one site but with multiple sites, maybe if there are mutations, they are functionally non-effective. What is extraordinary that because the protein has 82% in the dry weight, that there is one disease in which these patients have no loricrin and are still alive. So maybe that is the function. Or maybe Vovinkel is an extremely unusual situation in which there is an unusual compensation by other proteins. So maybe those proteins, they are not compatible with life mutations, and therefore we do not see it. So I think there are only speculation whether multiple sites, they are still allowing a flexible protein without the disease, and so I can only speculate in this sense. For the, what was the first question? I forgot.
No, the aging.
The aging. There's been reports on that P63 has some relation with aging. In theory, the skin should not be very much affected by aging, in theory, because every 30 days the skin is completely renewed. So there is no...
excuse me if I interrupt, but
this the
only
cutaneous,
yeah.
the lower layer. Yes, this is
The
the
lower
problem.
layer and the derma
Yeah,
is
yeah.
completely affected by
Completely
the
affected.
aging.
That is the hydration, and there are, if you measure the level of P63 in, we published that some time ago, in people below the age of 10 and 70, there is a huge difference into the expression of P63, and there is a huge difference. There is much lower in with aging, much lower level of expression in keratinocytes, and probably this changes the interaction with the subcutaneous tissue, hydration of the subcutaneous tissue. So, and there is a number of microRNA which are controlled by P63, and some of them, they are able to control the expression of P63, and therefore there are at least 5 or 6 microRNA which are control in both directions, P63, and they control the expression, the function, and the rate of proliferation of differentiation of the cells. So for the skin, although at the simplistic level, every 30 days is completely renewed, in reality there is an effect of aging. And you can use not only P63 but this microRNA to see how old is the skin, how much this interplay with the subcutaneous tissue for hydration, and therefore retention of fluid, and therefore elasticity of the lower side of the skin. That is not been fully studied, but yes, there is a relationship with the ovocytes. Is not my field of expertise, but I think we were referring to that Nature paper. There must be some way of controlling the efficacy of oocytes, and that could be of interest for the fertility clinic and for the control of the menopause and postmenopausal effect.
Thank you very much. I would like now to ask if the people online would like to make a question, if they have some comments, or if not, I open the discussion with all the people in this audience, and please make question. Caterina?
Gerry, my question is
Carolina.
about normal ovocytes.
Sorry.
Another Caterina. Normal ovocytes, not cancer ovocytes, that die because of ATM and CHEK2 and P63 epithelial. And my question is aimed to understand how and if external environmental factors such as hormone, for example, can influence this fate. So the question is there any physiological rescue pathway by kinase activity or by regulators of P63 expression during this sulfate?
The sulfate, it seems to be controlled by P63 and then kills. So the control could be only by blocking this, and therefore by blocking caspases, controlling like venetoclax on BCL2 function, or controlling the biphosphatase, or blocking the kinases, controlling the activation of P63, because once it's activated, the ovocytes are dead. So I don't think of having any regular medical treatment that touches this pathway for use at the moment.
And so hormones do not influence P63 expression?
I think the P63 is sensitive to the environment. Whether the abnormality is due by hormonal receptor activation, or physical toxicity, or chemical toxicity. Chemical could be, for example, an imbalance of ROS, regulated by hormones or by growth factors, by other things, this definitely will influence the ovocytes and therefore allow the premature chemotherapy is in cancer is a classic event of this, no? But probably also hormonal imbalance should be able to do this. So maybe why P63 is there, because when there is an abnormality, you do not need to have a transcription, expression of the protein, synthesis of the protein. You simply activate the protein is already there, and you have a conformational change which is less than 1 second, and then you kill the cell. So probably the necessity is the rapidity of a response to damage. That's why there is P63 and not P53.
Yeah.
P53 is lower in a way.
Thank you.
Caterina?
Yes, Gerry, very nice presentation and very nice overview of what you have, of few things that you have done in the past 10 or 20 years.
I'm old.
So I have a question regarding cell death in keratinocytes, because you touched on it, but you didn't go into details. And I was wondering about, for instance, so what do we know about cell death in the upper layers? And is caspase 14 in any way involved in that?
Caspase 14 is involved, but caspase 14 has nothing to do with apoptosis. And what is the function of caspase 14? That was cloned by Peter van den Abele in Ghent, but then he abandoned the field. So as far as I know, there is not many people working on caspase 14. Going on the role of cell death in the skin in general, I said certain things without any scientific fact. I assume that the fact is that sunburn cells, the dermatologists or pathologists, they call sunburn cells, these are apoptotic cells, they are only in the basal layer. They never see in the upper layer. And what is not substantiated by fact is that there is no evidence that you have P53 and classic apoptosis into the upper layer. There is no evidence whatsoever. Because there are no mitochondria, and a lot of the cell death mechanisms, they go through mitochondria, and there are no nuclei. No? So I guess in the upper layer, it's impossible to have a classic apoptosis, so classic mechanism of programmed cell death. But nobody has ever done it. That would be a very easy research to demonstrate that apoptosis only in the basal layer. You never have it in the upper layer. I don't know why. I've discussed it several times in my lab, but nobody wanted to do it. But I guess it's impossible to have, because you don't want death of a keratinocyte by apoptosis. You want transglutaminases, that cross-link, you want that substrate. Otherwise, doesn't work. So theoretically, it's impossible.
Okay, in the last, I have a comment more than a question. So regarding the P63 delta 13, exon delta 13, delta exon 13, first of all, the part on the oocytes is really fantastic, fascinating, because I mean, we really show that TAP63 alpha works in a certain way.
It was expected because Frank said it before, but we demonstrated.
Yeah, exactly. Very nice. Regarding the epidermis, as you probably know, we have the same. We are working on the same phenotype, on the same mouse. The 14 Cre delta 13 P63, and we get slightly different results. So we don't have any phenotype whatsoever of the skin. So it will be interesting to find out how
To compare.
to compare the two mouse models. We should talk about this.
It is possible, in science, this happens a lot. I was just mentioning that P53 was discovered in 1979 by Arne Levin, and the function was in 1989. It took 10 years to actually, and they only understand it because they discovered that Arne Levin was working on P53 and Varda Rotter and Moshe Oren, they're working on mutant P53. So one amino acid completely changed the stuff. So it is possible at the background of the mice or something gives a different phenotype.
Yeah.
It is absolutely possible.
Absolutely.
And therefore comparing, I think, is understandable.
be
So
very
we do have a clear phenotype. But like you say, they need to be compared.
Okay.
You have not published, we have not published either. So.
Okay.
On also with different results, we take some news with our, maybe there will be the final answer. So we are very glad that there is there is doubts about some results about sense.
Yeah.
Okay, we have to go to lunch, but there is I think some time for further question, if any. I would like to make a very big thanks to Professor, and thank you him very much for this visit.